So I'm Krista Niguchi. I'm a postdoc in Texas A&M, but this work is in collaboration with University of São Paulo in Brazil, and also North Carolina State University. But to start, I want to tell you actually a personal story, my story with sequencing data, more specifically with GPS data, genotyping by sequence. And I started to work with GPS data in 2015 in my master's. And I did my undergrad in biotechnology, so I have a biology background. And so I could understand the protocol, the wet lab part, like how to, we just sequence part of the genome, we use restriction enzymes to cut and just sequence around these cut sites, and how to include the adapters and how to include the barcodes to be able to sequence more than one samples, multiplex in a lane. And so this theory I knew, and I knew how to do like pipetting, and I knew like the small drops and transport drops, and it was fine. But then in my master's, I just received the data. So I needed to explore the bioinformatic parts. This part was a mystery for me. And I needed to learn many other things other than the pipetting. I needed to start to see these black screens with full of numbers and letters, and it was very hard to understand all the formats. So this is an example, a really small example of a passkey file and VCF file that if you work with markers, you probably already saw something like this. And I also needed to learn how to use computers that were much more powerful than mine, and learn how to manage the computational resources, like learn what's the difference between a core and a node, and how much RAM memory my procedures would take, because I would not be just using my computer and using the university computer, or sometimes the cloud environment. And with that, I needed to learn Linux and also how to survive without being the super user of the computer, like how to install all the softwares, all the dependencies without bugging anybody. And this could be really hard if you want to do the analysis of your GPS data, because there are many software available. And for each one of the steps, like for the quality control, we have many, and alignments, we also have many, and SNP calling, do the dosage calling or the genotype calling also. And we also need like have these other softwares for manipulate your files, your intermediary files, that can be really hard. And so with that, and all of them with different programming languages and different, sometimes just working specific operational system. And it's what was hard for me. And also like they are always passing through updates. So sometimes you write your code and then the developer decides to release another version, and then you want to update, you want the most, you don't want to be outdated, but then you update and you break your code. So it's can be really messy. And at the same time, my goal with the GPS data was to build a linkage map. So I needed to learn the theory behind how to build a linkage map in outcrossing population. At the time it was deployed locally, but it's still hard for me because in undergrad, I studied only like backcross, and it was easy with, it's not easy, but I knew how to do with backcross in red lines. So I needed to learn that we also need to estimate phases and how to use the Heiden-Markov model to estimate the genetic systems with multi-point approach. And what helped me was this package that was already there when I started, the One-Map package that it was made for build linkage maps in diploid species, and it can be used for inbred and also for outcrossing. And all the types of markers can be used like via biallelic and multiallelic or dominant and codominant markers. And it's writing R, so I started to learn R too. But so far for me, while the wet lab was like the small drops and pipettes, the bioinformatic for me was like numbers and black screen. I knew the theory, now I could, okay, I know what's happening, but I didn't actually believe in it. I just believed when I saw a graphic like this. When I saw a graphic like this, I say, okay, I'm doing something right. Like, oh, I could, okay, all the upstream step was correct, because this made a lot of sense to me. And this is a recombination fraction matrix. And the color of the graphic means the recombination fraction. So more to red means that the markers, the distant, let's start again. It's really important to you to understand this graphic, so I will spend a little time here. And so the red color means that there's not much recombination happening between two markers, and the markers are in the axis. So I order the markers by the genomic order and both X and Y axis. And so closer they are from each other in linkage, they should be more red. And if they have many recombination between them, it should be more blue. And I got, so you can see that's two different sources of information. Is the genomic order that's from the assembly, and the linkage of the number of recombination that comes from the genetic, my population. So if I see a pattern of color like this, the red on the diagonal and gradient coming to be blue, it means that the order of the markers in my genetic population match the genomic assembly. So it's two different sources that comes together and makes sense in these beautiful colors. And then say, okay, nice. And this was my actually first graphic. So it's a historical thing here, and has some issues with this graphic after I fixed it, but I wanted to be faithful to my story. So I put the original here. And my data was I was working with eucalyptus at the time. So with the same logic, also if we have like differences between the genetics of the population and the genomic assembly, I can also see in this type of graphic. So in this graphic here, you can see that there's a break in the pattern of the color that I expect here. And then what I did, I came and I took this block of markers and I flip it. So I just corrected the inversion that there is the difference between the assembly and my population. So I could even detect the inversions of the inversion comparing to the assembly. So some of the difference of the pattern that I'm expecting with what I'm seeing, I can explain biologically, but I could find others that I look like, this is weird. Like this inversion, translocation, what is this? And actually they're just like bad data. I cannot explain that because I cannot see the gradient very clear from red to blue. And also I have like these blue lines that doesn't mean anything. It's just like, okay, bad data. And then I start to complain about OneMap because it's not working. It's like, it's giving me a map that's not supposed to. And also OneMap was released in 2007. So we're not expected too many markers, as sequence markers we usually provide like thousands of markers. So the analysis were too slow. And even when I had a recombination fraction matrix that was nice, the map was still inflated. So I was like, okay, not working. And then I learned that be careful with what you complain to, because then I started to develop, I joined the team of developers of the OneMap and became the maintainer in 2017. And I started to do updates. So we recently released the 3.0 version. And luckily, while I was having this issue, Marcelo was also developing a much more complex package, that's the MapPoly, because it's able to do linkage mapping polyploid species also. And I will not talk too much about this because I don't want to give spoilers. They have a talk later, so just watch their talks. They also have updates on MapPoly. But we were talking and we were having the same issues, like sometimes the data, we had like these genotyping errors and we didn't know what to do with this inside our package, OneMap and MapPoly. We realized that if we have markers that are not good, there's not much that we can do in our side. Like we provide bad maps because the markers are bad. So I started to explore all the steps before the package, before building the map, and trying to provide the best markers possible without genotyping errors. So what I did was I changed the combinations of softwares before. So the basically the SNP calling and the genotype calling and those or dosage calling. And I also changed parameters to see what happens with the quality of the markers. And so I use GATK to do the SNP calling and freebase. And I use also GATK markers, but the polyred dosage, and also updog and freebase polyred and freebase updog. As you can see, different combinations of software provides different quality. And now here I'm using the heat maps because I hope you also have like the same feeling with the other heat map, because for me it makes sense. So if the heat map doesn't make sense, it means that there's something wrong with the data. So this would be an example, this one here. It's an example of a heat map that I don't want because there's a lot of blue lines where it's not supposed to. And this here is an example of heat map it's okay. And you are not seeing too much blue in the edges because this is just a subset. Like I took only 37% of the chromosome 1 of a data set of diploid roses. And so it's just a block. It's not expecting too distance of markers. And this data set is very good. It's 9.4x. And all this was built in one map. And I did just a subset because I did a very intensive study. I built many maps because I wanted to test many things. So basically I did with this empirical data, the diploid, about 800 maps. And I did also simulation study, and then I did like 10,000 maps because I wanted to test many things. And doing that, I could detect like, okay, this software is making a lot of mistakes in this situation. So I created like filters to avoid that the software make mistakes. And I could get better maps after that. So this is the heat maps before the filters that we use it, and this is the heat maps after. So you can see that there was an improvement. And but even doing that, the maps were still inflated. Like this subset, I would expect to have like 38 centimorgans, but I end up getting like 540. So something was still wrong. And the issue was not that just the map is inflated, but with the map inflated, it means that we have too much recombination where not supposed to. So we estimate also the haplotypes of the protein, that this is the goal of doing that. So if you see like this 6 individuals example, you can see that we have some lines here. That means that double recombination happened very close to each other, what we don't expect. And then it's okay, we still have many errors. But luckily, we work with Heidenmarkov model. The multi-point approach we have a way to use more the genetic information instead of the information that's coming from the markers. So I will try to explain this. I know that can be hard, but Jiken did a very good graphic that I consider very didactic about this, because when you use the Heidenmarkov model, it will use the multi-point approach to sometimes change. If you allow it, it can change the genotype or the dosage according to the multi-point information and not the genotype that you give. So the genotype that is the result of your analysis, not necessarily the one that you go in, the input. So this graphic that Jiken did, he compared the dosage that you receive with the dosage that after the Markov model. And all the black, it's genotypes that were not changed. And the green ones are the imputed, of course, because if you have missing data, the Heidenmarkov model you input according to the chain. And but the red ones was the one that the Heidenmarkov model changed it, because in this case, he allowed 5% of error. So instead of you giving just a value, you also give a probability of this value to be wrong. And you give like this a flexibility to the chain to change your genotype. And you can use like just a value, like he did in this case, like just 5%, Or you can use the probability that comes from the upstream softwares, because the software that do the dosage and the or genotype calling, as you can call, it provides some probabilities for you. So you can use this in the Heider-Markov model. And I also test this, like what happened if I use a global, or if I use a really small, if I give a small flexibility to the chain, if I use the upstream probabilities, or if I use a 5%. And I took a heat map that I considered good and was still inflated, and using the probability of the softwares that coming from the upstream softwares, it reduced a little bit the size of the map, but still not as much as we expect. But using the 5% reduced a lot. So it comes very close to what we expect, that would be the 38. And then, and this changed the size, but also changed the haplotypes, of course. Like we don't have much more of the double recombination that we had before. So this is, okay, this is make more sense. And so we start to ask, okay, we know that inflated maps, it's not right, but it's small maps always good? Like, could we use just this as a metric as our map is good or not? And then because we have the simulations, we did the simulation since the simulation of the sequence, we got all these maps that we simulated. So each dot in this graphic, it's a map. It's that map with just 37% of the chromosome. And we measure, like the x-axis here, measure the difference between, is a measurement for the difference between the true map size and the estimated map size. And the this is the y-axis, and the x-axis measure if the wrong recombination breakpoints. If the breakpoint is where it's supposed to be in the haplotypes. So here I would expect that to be here. If the break is not here, I would consider wrong. And if there is a break where it's not supposed to, I will consider wrong too. So underestimated and overestimated is considered in the graphic. So, and then each block is, here's a small error rate, so few flexibility, and 5% of error, or the relative errors, the error coming from the upstream softwares. So you can see that if I use 5%, I can have maps that are small. So this is the square, the red square here, that's meaning. So I have like less than 1 centimeter of difference of what's supposed to, but still I can have from 10 to 100 wrong recombination. So yes, it's good to have small maps, but if still you have a lot of genotyping errors, what can happen is that you estimate recombinations where not supposed to, and you don't estimate recombination where it's supposed to. So you can have both under and over, so you cancel each other and the map is correct, but the haplotypes are wrong. So I would be careful to increase too much the error rate, the probability that you put too much flexibility to the chain. And we could do that because we have the simulations, and because we have the simulations, we test other things like segregation distortion. What happened with the maps if we have the segregation distortion? What happened with the maps if we have contaminant samples? And what happened with the maps if we have multi-allelic markers? So I will not discuss all of this, but we have a preprint available already with all these results if you want to check more. And we didn't this also not with a single data set, but we changed it. We use another data set with other characteristics. So this is a Diploid Aspen data set. And also I did the same thing. Oh, sorry. I did the same thing. I just used 37% of the chromosome 10. And this data set is very different because there's no so high depth. This is only 6x. And I can see a lot of difference between these heat maps and the heat maps that we saw before. And once, because yes, we have more blue, we have more errors in this data set. So the distance between them is higher, even if I have just a subset. And the softwares, the combinations of softwares doesn't give me the same results. So I cannot say, okay, this was best in the other data set, so it's best here. So if I change this data set and the characteristics of the data set, the combinations of software that brings me the best heat map changed. So I was very disappointed because I wanted a simple answer. Like, okay, I want to tell everybody use this and this software, but I couldn't because different data sets give me different results. And then recently I changed, we also did this for tetraploids, for polyploids. So instead of using one map as the software to build the linkage map, I'm using now MapPoly. And I saw the same thing, like different softwares, different parameters give me different answers. And so this is the tetraploid rows, and this is the entire chromosome, the results for the entire chromosome. So I have good and bad heat maps. And for the polyploids, I also did the same thing. So I applied 0 as I don't allow the chain to any flexibility, so applying 0 as error. And I also use the upstream software probabilities and a 5%. So I can see the same pattern as diploid. So 5% is usually better. And okay, you will see many maps. So I show you the results first. And because the methods for doing that was very hard, because I told you at the beginning how hard it is to deal with a lot of softwares. And I'm not doing only one software. I'm not using only one map and MapPoly. I'm doing since the sequence. So I'm going through all the steps to all the map that I show you. And this can be hard. So I have these issues and I needed to, these technical issues that I needed to solve. And one of them, like dealing with large software, was okay. I have the HPC's computers, and I also have like Google Cloud or Amazon Cloud if I need for the resources. And for the many softwares, the many programming languages, and to deal with the updates, I started to use containers. That was very useful for me. And these containers, like briefly, what they do is isolated your system, the system that you run in your analysis from all the others. So you keeping this isolated system, your operational system, all the dependencies in the software that you are using. And this is keep it frozen. So you can, all the updates that happens around will not affect your analysis. You just change the versions when you want. So this was very useful. And there are like repositories that I can find containers that are already published by the developer. So it's very easy. So I replace all the softwares by just using Docker or Singularity. That it's the softwares that you use for accessing the containers. And then other technical difficulty is that I have many steps since the sequence. So I have all these and have like the intermediary files that I need to handle also, and can be hard. But I solved that using workflow systems. And there are many available, like Galaxy is one of them, Nextflow, SnakeMake, and CWL, and the WDL, also known as Widow. That was the one that I explored and I use in this work. And there are also repositories available for workflows. So if you want to do other analysis, and it's a good idea to look to these repositories and see if someone did before, and you can just reproduce what they did or change some stuff. And these workflows allow you to run in HPC and also in cloud systems. So I replace all my Bash independent files by using workflows. And now, so I built the Reads2Map using the Widow. So I like this because this system, because it's very modular. So the smaller unit, it's the tasks, and I can take the task, combine the task and create a sub workflow. And I can combine sub workflows and create a workflow or a pipeline. And this is the Reads2Map repository. So it's distributed in these directories if you want to access this. And if you want to run a workflow, just the pipeline, it's pretty easy. You can just create the input file. The input file, you need to tell like what you want or what you don't want to run, and the path for your input files. That's for the Reads2Map is the FASTQ files, a file with a list of your FASTQ files and the reference genome. And you can use the TerraBuild. TerraBuild is the cloud environment. It's a graphical interface that Broad Institute did, was very good. for running it. You can like have buttons to input and to run the analysis, or you can use the HPC systems and run with Cromwell, MidWDL, and the XWDL. And with a single line, this is with Cromwell, so you can specify your inputs and specify the workflow that you want to run, and this will run all the analysis for you, and you just get the outputs. But doing that, you use all the parameters that I defined for my data sets, and cannot be the best ones for your data set. So if you want to change the parameters, then you need to explore a little bit of the tests. And I have many tests because I have been working with that for a long time, but I try to split it by the name of the software. So if you want, you can access this in the directories. And I split here to show just the freebase one, but I think I'm running out of time, so I would skip it. But I just show the structure of each one of the tests. It's split between inputs, the command, that it's the place that you should change your parameters, and the runtime, that's where you specify your container, and the output, that's the file that will output. And you will combine the tests, just connecting the inputs of one test by the outputs of the other. So this is other example. I will not explain, but it's there if you want to see, and you can talk to me later also. Okay. And this is the summary of the workflow. So all these tests that I just showed is to do that. Like you go in with your FASTQ files and your reference, and you run alignment with BWA and SNP calling with GATK and freebase, and genotype calling with updog, polyred, or supermassa, and do the linkage map with 1map or mappoly. And as a summary, we can see that building a linkage map is very sensitive to the upstream choices. And I could not find a single suggestion for all data sets that I tried. And the Reads2Map, it has the parameters, the default parameters according to the vignettes that I wrote, that I read, and everything. I tried to keep the default, but I asked sorry to the developers if I didn't use the correct parameters. But if you can let me know, it's perfect because I can change this very easily. And I would love to have better maps. And the Reads2Map is this organized, this structure. Then it's very reproducible and can be scalable. Like I built many maps, and it's easy to change. So with that, I want to thank you, everybody that helped me with this work. And that's it.